Hello and welcome to Remote Lesson 2. This is on experimental design. By the end of this session, I'd like you to feel confident that you understand the key experimental terminology, that you can apply your understanding to design experimental methodologies, and also that you understand the difference between systematic and random sampling. So before we start with more sort of descriptions and stuff, I'd like you just to pause the video where it's at and to go to the Biology VLE, the Devil's Dyke Fieldwork folder and the Remote Lesson folder, and there is an activity in there called Experimental Design, and I'd like you to complete part 1 of that activity. So please pause the video and go and complete that activity. Okay, so we're going to review the first part of this activity by doing a few sort of quiz questions on this. So you'll need a pen and paper or whiteboard, whatever you're using at home. So how this next part is going to work is that I'm going to show you a few images, kind of like this one here, and I'd like you to identify the key term that links to each of those images. I've actually said on each one how many key terms link in. So for example, here there is one key term that links, and the clue is there will be no significant difference between the number of snails in area 1 compared to area 2. And so the answer here is that is a null hypothesis. Hopefully you remember from the video, as part of that activity that you've just done, a null hypothesis is where you say there's no significant difference or correlation between your two groups or your two variables. So here we can see there's no significant difference between our two groups, and therefore we would accept the null hypothesis, but that would have to be after we've done a stats test. Again, more on that next week. So as we go through, write down the keywords that you think link, and then we'll see if you're right. So here's your second question, or clue. There will be a significant correlation between the number of ice cream sold and the number of shark attacks. I predict there will be more shark attacks when more ice creams are sold. So there are two key phrases or key terms linked to this clue. So feel free to pause the video, but if you could just write down which ones you think they are. And here are the answers. So this is an alternative hypothesis because we're saying that we predict a significant correlation or difference between the IV and DV. And actually this is a one-tailed directional alternative hypothesis because we're saying that we're predicting a positive correlation. So as more ice creams are sold, there are more shark attacks. And this is actually also a true data set, but again sort of links in with causation versus correlation. Okay, so you can think of, is it causation or is it just a correlation? Clue number 3. So hopefully we identified that that is the independent variable because it's on the x-axis. Clue number 4. So again, hopefully you identified that this is the dependent variable because it's on the y-axis. Okay, there's 3 key terms for this picture here, so see if you can spot them. And hopefully we identified that we've got in every single practical, as you guys know, you have to have an independent variable, the thing that you're changing, a dependent variable, the thing that you're measuring, and then other controlled variables. So just make sure that you're still writing it as controlled variables, not just control variables, and that would provide the sort of basis for our practicals. So here you guys might recognise sort of that we used similar equipment when we did the enzyme practicals. So we used catalase, the enzyme, and catalase breaks down hydrogen peroxide into water and also oxygen. Okay, what is the key term here? And hopefully you've identified that this is the standard deviation. So again, how much does our data differ from the mean? What is the key term here? And hopefully you've identified here that we're looking at reliability. So you might remember from the last video the smaller the range or standard deviation bar, whatever's plotted, so let's imagine it's the range, the smaller the range bar, the more reliable the data, the larger the range bar, the less reliable the data. And here we can see we are reliably inaccurate. Okay, what is the key term here? And you might recognise that this equation is the standard deviation equation. So do make sure that you feel familiar with using this. Again, all of you will have used this at GCSE, but you might just need to remind yourself and just go back over it. There's two key phrases here. And hopefully we identified that that is difference and correlation. So we have to be careful if we're looking for correlation in our data, then remember we're going to be plotting a line chart as this one shows here. If we're looking for a difference, we're going to be plotting a bar chart. And we're going to be using different stats tests depending on what kind of data we're collecting. So for a correlation, we're going to be using Spearman's rank correlation coefficient. For looking at a significant difference, we're going to be using the student's t-test. Okay, so if you're ever not sure what stats test you're going to be using on a data set, just plot a quick or sketch a quick graph of what you think your data is going to look like. Generally, you really need to think about the independent variables. So you're going to be plotting a bar chart if you've got a continuous independent variable, for example, something like height or length, whereas you're going to be plotting a bar chart if you've got categoric data. So let's say we're comparing male cats or whatever with female cats. Okay, so two different categories. Okay. What is the key term linked here? And hopefully we've identified that this is accuracy. So we have been very accurate with firing the dart here. We've hit the bullseye, and we've got a more precise unit at the bottom here. Centimetres is more precise because it's smaller than inches. And so if we use centimetres to measure the length of this leaf, if we're measuring to the nearest whole centimetre, so that's going to give us a more accurate reading compared to if we measured to the nearest inch. Please can you go back to your experimental design activity. I'd like you to complete part 2, pause the video, and then we'll carry on with looking at the answers. Okay, so to look at some of the answers here then, you should have watched a short video about designing a methodology, and as you guys are all doing biology and probably other sciences as well, you will be naturally scientific thinkers, so you'll be quite familiar with the scientific process. But just a reminder that it's not a completely regimented process. It can be quite fluid. So you might yourselves have made, or often people make observations and then they think about why those observations are potentially occurring, and so then they might design, a scientific thinker will design an experiment around that to sort of test that, and they'll develop hypotheses and stuff like that. So again, the hypotheses, that links in with what we were talking about earlier with developing a null hypothesis. The null hypothesis you're testing in your statistical analysis. Okay, so you're basically testing to accept or reject your null hypothesis when you do your stats analysis. So when you're planning your investigation, you do always have to think ahead of how you're going to have to statistically analyse your data, and that will help you decide on things like your number of data sets, so your range of your IV, your number of repeats, and so on. Okay. So you had 2 questions in your activity, and you guys were asked to identify these key features. So we're going to look at those for each one, and we're also going to link it with the idea of random and systematic sampling. So let's start with question 1, which is the species richness of a plant, from the low tide shoreline to the tree line of a beach. So this question, the first thing that we want to think about when we're looking at ecology, and again, some of you might be doing environmental science, so you might go into this in a lot more depth. We are looking at it in a very, very basic and very, very crude way. So please do bear in mind that there are so many different ways to test things in ecology, but we're looking at just a couple. But if you are interested, please do read up. There's extra resources on the VLE about if you can sign up to the environmental science page, then you can maybe get a bit extra in there. So very crudely then, this question, whenever we're looking at in biology, whenever we're looking at an ecological gradient, we want to be thinking, about using systematic sampling. Okay, so an ecological gradient is basically in ecology where you've got a change from one distance to another distance. So, for example, here we've got a change in the ecosystem or in the sort of spread of species from this point here all the way up to this point here. And in the question, it said that while you were walking up the beach, you observed that actually the species richness changed as you moved away from the shoreline. So a lot of you will probably have identified that that's probably something to do with the salinity, so the saltiness of the sand, and the ability of plants to kind of cope and deal with that. Salty environments are very, very hostile, so you don't get loads and loads of plants and things like that growing in those environments because they require very, very hardy adaptations. So you might also develop at this point a null and alternative hypothesis, okay? And so you might be thinking, all right, well, I'm going to come up and develop a one-tailed alternative directional hypothesis where as I walk away from the sea up the shoreline, I notice that the species richness increases. So maybe we've got, let's say, from point X to point Y, point X to point J, for example. Along this transect, I'm going to plot it on my graph, for example, here. So from point X to point J, up here, I can see that my species richness, which will be on my y-axis, increases. So I've predicted a positive correlation. And this is described as systematic sampling. Okay, now systematic sampling is basically where, or often where you're using a transect. Okay, so it occurs across an ecological gradient like the one that we were just looking at on the beach. And what we can do here is if we're looking at an ecological gradient, we can set up a transect. It's basically you'll pull generally like a measuring tape along the ecological gradient, so for example here, and then at certain points along that transect, so that measuring tape, you will take certain points where you'll be taking measurements. Okay, so maybe every 2 meters, for example, and you can see this scientist here has stopped at this second reading point and they've placed down a quadrat. And so you potentially have used this at school before, but here's a quadrat at the bottom. And quadrats are used to measure plant abundance, distribution, species richness. Okay, you can't use these to measure insects or anything that's moving. And also anything that's, you couldn't use a quadrat to measure plant, like massive trees, unless it was a much, much larger quadrat. So this small one here, for example, we couldn't use on everything. We have to use the right kind of quadrat for the right kind of organisms. So within this quadrat, we can see here that our species richness is 1, 2, 3, just from the looks of it. So we've got 3 different species. Or we might be even better, even more specific, and we might measure the species evenness. So we'd take into account the number of individuals within each species as well. That would probably be a much better way of measuring the biodiversity. So that's systematic sampling, setting up a transect along a gradient. Now do bear in mind that, again, you don't have to necessarily use a quadrat if it's something like an insect population that you're measuring. Generally, you'd have a much, much longer transect. You couldn't imagine that different populations would occur so close to each other. They'd probably still be part of the same population, so you could set up a much, much longer transect and drive between points. But you could sample your insects, for example, by using things like sweep nets or pouters, and so these are other ways of collecting those insects, identifying them, and then recording them. So if we link that back to the activity, here are the answers for it. So you might want to pause the video and just to check over and see if you've identified all of those things correctly. So just one or two things to point out here. The first one is when you're identifying your independent and dependent variable, be as specific as possible with exactly what you're measuring and exactly what you're changing. So the independent variable, we were measuring from the low tide shoreline. So we're measuring the distance along that transect. The dependent variable was the species richness. So, if you're measuring abundance or percentage cover, whatever it is, you have to be really specific with that. Controlled variables, so again, whenever you're describing practicals in exam questions, just one thing to highlight is that, yeah, you can identify your IV, DV generally quite easily and also your units, but you do also want to give at least 2, probably 3 to 4 different controlled variables, and again, ranges and ways of kind of monitoring them depending on how the question's phrased. Again, with all practicals, you want to think about these things, but with this one in particular, so the species richness of plants from low tide shoreline to the tree line of the beach, things that we might consider for the controlled variables are we going back at the same time of year, same time of day, same tide time? Maybe the areas that we're measuring in, we might want to just check the pH and just check that hasn't changed. Maybe there's some sort of effluence pipe or whatever near the shore that might change the pH of that ecosystem. The gradient and of that beach, and also the aspect, so which directions are they facing? Are you measuring on more than one beach? It's really, really hard in ecology to regulate these factors because obviously you're outside of the lab. If you're in the lab, you could regulate the light intensity and so on and so forth. So you just have to be mindful, and you just have to try to regulate and keep these things as similar as possible. So go back the same time of year, try and make sure it's the same time of day, and so on. Methodology, so as we said, this is a form of systematic sampling that we're going to use here. So we're going to set up a line transect that spans that beach, and because it's a line transect, the data that we're going to be getting is going to be plotted on a line chart. Okay, so we're going to be looking at a correlation. So for Spearman's rank correlation coefficient, again, you do more next week, you need 7 or more intervals along that transect. Okay, so between sort of 7 and 30 generally. And you might place a quadrat at each of the distances that you've identified, so every 2 meters or whatever it is. Again, you'd change that depending on the ecosystem. So it might be that you take samples every 2 meters in one ecosystem, but that might not be appropriate for another because maybe the other ecosystem is much, much shorter. Reliability, whenever you think about reliability, repeat. R for reliability, R for repeat. And our null hypothesis is there's no statistically significant correlation. So there's our keyword. Make sure that you're careful with that. We're not looking for a statistically significant difference. It's an ecological gradient, therefore it's a continuous scale, and therefore it's a correlation. We're going to be plotting a bar chart like we said down here. Alternative is we predict there's going to be a positive correlation as we move up the beach. The number of species is going to also increase, and we can use Spearman's rank correlation coefficient to measure that. Okay, random sampling. So question 2, the influences of soil pH on species richness of plants in 2 different fields. So let's imagine we've got field A and field B, and they're separated by a river. And you went for a walk and you noticed that actually it looked like the species richness was different in field A compared to field B. So here, again, straight away when you're doing this, think about what data you're going to be collecting. So we're comparing 2 different fields, 2 different sites. So we're not going to be plotting a line chart. We're going to be plotting a bar chart because we've got 2 different categories. Okay, so field A and field B. So therefore, that shows us that we're going to be using the student's t-test because we're looking to compare if these means are significantly different. Okay, so again, straight away, we can identify the kind of data that we're collecting and the sort of stats test that we're going to be using. So random sampling is what we're going to use, and this is basically to get a representative sample of a larger area, so something like a field. So here we don't have an ecological gradient, we're just comparing 2 areas. So we can go into our field, and the first thing that we want to do is to set up a grid that we're going to take measurements for. The easiest way to do that is to run a couple of tape measures, so you've got a grid kind of like this. You can then use things like random number charts or generators, so you can often find them on Google. So for example, here in my random number generator, I've said I want numbers between 1 and 100, and when I click generate, it gives me a random number. I could then click and get another random number, and let's say my second random number was 52, for example. Sorry, my writing's not very good. And so my first number here, number 42, I'm going to imagine that one I walk along until I get to 42 meters along my x-axis, and then I'm going to walk up until I get to 52 meters on my y-axis, and then at that point there, that is where I'm going to put my quadrat. Okay, I can place my quadrat, and I can then count the species richness. Okay, I would then repeat this a number of times, so generally I'm going to be repeating this for the students' t-test 6 times, and then I've got 6 data pieces for that particular area, and they're all random. So it's, you know, one of the things that people often think with quadrats, I'll just go and I'll just chuck it in the field, but that is generating a bias, because you might chuck it in a certain direction because of some unconscious bias that you have. So we do have to be really careful to make sure that it's a random sample. So answers to that sheet then are independent variable here, so the pH of the soil. We've got 2 different fields, so field A and field B, and maybe we have identified that field A has got a certain pH of the soil compared to field B. Dependent variables, so the species richness is what we're going to be measuring. Controlled variables, so again, because it's in ecology, it's a little bit hard to control them, but you can account for them, so you can make sure that you're going back again same time of year, same time of day. Has there been a similar amount of rainfall, because potentially that would affect things like the pH of the soil. And other influences, so things like farms, for example, or whatever nearby. Methods and techniques, so again, you want to identify an area within the field. You're going to create a grid, and then using a random number generator, we want to identify coordinates for 6 different locations. We can then use our quadrat and count the species richness at each of those locations. Again, reliability, this is always the same whenever you're asked about this. I carry out repeats, I calculate the mean, I maybe check for outliers. Our null hypothesis, so remember with this one, we plotted the data that we were looking to see, and we were comparing field A with field B. So we were looking at a bar chart. So again, we're looking at, is there a statistically significant difference, not correlation, so be careful again. So our null hypothesis is there is no statistically significant difference. Again, that's what our students' t-test will be testing for. And then we can accept or reject it. Our null hypothesis. Our alternative hypothesis is saying that there is a significant difference between field A, the species richness in field A, and field B. Again, student t-test, that's our stats test. So hopefully you guys feel confident about the experimental terms. Hopefully you feel like you can apply it to design methodologies, and also hopefully you feel confident that you know the difference between systematic and random sampling. Okay, thank you.